네, 그러면 시간이 거의 다 돼서 오후 세션을 시작하도록 하겠습니다. 점심은 맛있게 잘 드셨나요? 오늘 오후 세션의 첫 번째 인사이시면서 두 번째 플래너리 스피커인 유지 교수님을 소개드리겠습니다. 유지 교수님께서는 89년에 고려대 의과대학을 졸업하셨고요. 94년에 동대학에서 박사학위를 받으셨습니다. 이후에 보스턴 칠드런스 하스피를 하고 하버드 의과대학에서 박사 후 연구원을 거치셨고요. 2001년에 하버드 대학교 비뇨기과의 조교수로 임명되셨습니다. 2007년 이후부터 현재까지는 웨이크 포레스트 유니버시티의 교수이시면서 웨이크 포레스트 인스티튜트 for Regenerative Medicine의 어소시에이트 디렉터로 재직하고 계십니다. 많은 상을 받으셨는데요. 그중에 R&D 엑셀런스 어워드 바디 온 칩이라는 토픽으로 2019년에 받으셨고요. 2021년에는 나사 베스큘러 티슈 챌린지 상도 받으셨습니다. 오늘 발표하실 주제는 translational of the Regenerative Medicine Therapy에 있어서의 문제점과 그에 대한 해결 방안에 관한 말씀이신데요. 오늘 미국 계셔가지고 발표를 바로 온라인으로 하시지는 못하고 프리레코드 발표를 전달드리도록 하겠습니다. 자, 그러면 유지 교수님 발표를 듣도록 하겠습니다.
Good afternoon. First, I would like to thank Dr. Dongwoo Cho and Dr. Gina Zhang and the organizing team at PostTech for including me in this symposium. Today, I would like to share our experience in translating regenerative medicine technologies and discuss what progress we have made. I would also like to discuss some of the challenges we're facing and what approaches we're taking to address these challenges. In addition, I would like to share our thoughts on how we could accelerate the translational processes. Regenerative medicine is a broad field that covers many scientific areas, including cell therapy, tissue engineering, developing diagnostic platforms, and support technologies such as bioreactors and delivery tools. While the approaches and strategies involved in each area may vary, the overall goal is directed toward developing effective therapies and technologies for translational applications. As you know, translational research path is a long journey that involves complex steps, starting with the research and discovery phase to all the way to clinical trials and commercialization. As you could imagine, developing and translating new therapies requires time, effort, and resources, but the upside of this effort is that it would benefit patients in need. Over the years, we have learned from our experiences, which has led to a set of questions for developing a therapy or product. These questions have served as a guide to ensure that the path we are taking is the right one that could result in a successful outcome. The first and foremost question is, does your technology or product, when completed, solve a clinical need or problem? If yes, then you may want to ask how it compares with the existing therapies. Is it better? Is it simpler? Or is it more economical? What is the benefit over the existing approaches? It is also important to know how you envision your technology to work. One critical question is, can your technology be developed in a timely manner? This is important because if your technology requires about 10 years to develop, could there be a new and better technology that are developed in a shorter period of time and make your product obsolete? Another important question is, can your technology be marketable and commercialized? Some products may be unsuitable for the market due to various reasons, such as high costs with mediocre outcomes. In this case, the highly invested products may not see the light in the market. Thus, it will not be distributed into the broad market. These are all important questions that must be answered affirmatively before any project with translational product in mind can be initiated. I would like to share our experience with you that involves the development of engineered bladder tissue. Bladder was one of the first organs we had worked on, and we started with a clinical need. There are many conditions in urology where additional bladder tissue is needed for reconstruction. In such case, bowel has been used to reconstruct the bladder tissue, as no other substitute tissue is available. However, the use of intestinal segment results in complications such as metabolic derangements, stone formation, infection, inflammation, and sometimes cancer development. Unfortunately, this is the only method available to treat many pathologic bladder conditions. So our approach to address this clinical need was to apply a tissue engineering strategy that involves the use of cells, biomaterials, and biological factors to build tissues for bladder reconstruction. The idea was to use patient cells combined with a biomaterial scaffold to build an implantable tissue construct that matures into a target tissue upon introduction in the body, in this case, the bladder. In a real-life scenario, when a patient who requires bladder tissue reconstruction is identified, a small bladder tissue biopsy is taken. Bladder cells are then isolated from the tissue and expanded in large amounts outside the body. When sufficient amount of cells are obtained, they are placed on a bladder-shaped scaffold that slowly degrades over time in our body. The cell-containing construct is then used to augment the same patient's bladder where the cells came from. The cells are then matured into tissue as the biomaterial scaffold degrades slowly in the body, and the patient ends up with bladder tissue consisting of his or her own cells. The tissue is intended to remain in the body as long as the patient lives. Following this approach, our initial clinical trial involved treating 7 pediatric patients with a congenital anomaly called spina bifida. These patients have an abnormal condition called neurogenic bladders that often results in kidney failure due to urine refluxing back to the kidney. Following the technology platform we developed, the patients received engineered bladder tissue with their own cells. The patients were followed for over 15 years. These patients showed a larger bladder size and stable low bladder pressure that does not reflux back to damage the kidneys. Following our initial trial, 2 independent phase 2 clinical studies were performed in spinal cord injury patients with bladder dysfunction and neurogenic bladder patients. We now have expanded the indication to treat adult patients with scar-contracted small bladders in a phase 2 clinical trial. This trial is supported by the Department of Defense through the Armed Forces Institute of Regenerative Medicine program. In this project, we aim to establish the manufacturing processes and develop standard operating procedures, or SOPs, for treating adult patients. The SOPs are validated and used to manufacture bladder tissue constructs to obtain FDA approval for a clinical trial. The SOPs for each procedure were developed through the process development, followed by process optimization and qualification in a GMP facility. We have developed a clinical manufacturing process specifically for the bladder tissue construct. The tissue starts with biopsy collection and transport to our manufacturing development center. The tissue is then dissociated and the urothelial and smooth muscle cells are isolated and cultured. The biomaterial scaffolds are prepared for cell seeding, followed by packaging and transporting back to clinical sites for treatment. On each step of the procedures, in-process testing is performed to ensure that all the elements are processed properly. We perform these testing when they arrive at the GMP facility, during and after cell culture to make sure they're not contaminated. And then during scaffold preparation process. We also perform testing on the cell-seeded scaffolds and finally perform the release testing prior to sending off to the clinical site. These steps are very complex and every procedure is recorded. Following the developed protocols for manufacturing, we have performed cell characterization, scaffold optimization, scaffold manufacturing, cell seeding, confirming cell distribution on the scaffold, as well as the media conditions in our GMP facility as shown on this slide. We're fortunate to have an established manufacturing development center at our institute. WFirm has all the teams and components necessary to bring benchtop research to clinical products. The center has a translational team that performs preclinical IND studies, process development team that turns research protocols to standard operating procedures, a quality control and assurance team, as well as the manufacturing team that produces treatment articles. We also have individuals that deal with regulatory affairs, and these capabilities support translating laboratory concept studies all the way to patient care. Besides the bladder application, the same strategy was also applied in 5 pediatric patients with traumatic urethral strictures, which is a narrowing or blockage of the urethra. These patients could not urinate before the operation, but now they are able to void after the surgery and throughout the follow-up periods. One very exciting outcome of engineered tissues is in the vaginal tissue application, where we had treated 4 patients born with abnormal vaginal anatomy. In this trial, patients' cells were obtained from their incomplete vaginal tissues, and these cells were used to create a new vaginal bulb. The reconstructed tissues were functional, and the patients have had normal sexual behavior after the surgery. Some of these patients got married and are living a normal life. And this is exciting because this strategy not only replaces damaged tissues, but allows to create a new organ for patients to improve the quality of their lives. These applications are currently being expanded to other clinical indications that would benefit more patients. We have extended the urethral applications to treat adult patients with strictures currently. We have just started a clinical trial involving the corporal tissue repair in patients with irreversibly damaged penile corpora, such as Peyronie's disease. In this trial, donor corporal tissue is obtained and processed to remove all the cellular components, leaving the extracellular matrix structures intact. Then the corporal structures are seeded with patients' own cells to repair corporal tissue defects. We're excited to apply these tissue engineering concepts to benefit patients. So our experience with these tissue engineering applications indicate that once a specific technology platform is established, the platform could be used with modifications for other tissue applications. I often get this question: when will these applications be available for clinical use? Of course, there has been a great progress in the development of engineered tissues and organs. This slide depicts the steps and approximate time required to develop and translate a product into the clinic, which may take about 20 years. That is, if every step is assumed to be successful. In the case of bladder application, it took us 4 years to obtain approval to conduct a phase 2 clinical trial, although it was an expanded indication for adults. Therefore, bringing technologies to the bedside is a long journey that requires patience. The development and translation processes consist of many steps, starting with the scientific and discovery phase. There are many scientific and development challenges that need to be addressed and overcome, which I will touch on it later. In addition to the scientific quest, there are steps involving process development, validation, and manufacturing that need to be sorted out, such as technology transfer and scale up and implementation of the developed processes. Furthermore, working out the regulatory issues and commercialization strategies are other challenges or opportunities involved in developing tissue engineered medical products, as shown on this table. Of the many challenges present in the translational processes, I would like to discuss some of the major scientific and technological challenges we need to address in building complex tissues and organ systems. And these include vascularization, innervation, development of tissue and organ-specific biomaterials, establishing a universal cell source that could be used across all patient populations without having to worry about rejection, and finding effective methods to deliver cells and tissues to target sites, and methods to enhance tissue maturation and integration in the body. Therefore, our research efforts have been primarily focused on addressing these challenges. One challenge I would like to bring to your attention is the engineering challenge that involves scaffold development and fabrication. The tissue applications I shared with you in the previous slides have been produced manually. However, it is impossible to fabricate the inner microtissue structures of complex tissues and organs by hand. We currently do not have the technology that can replicate the complex vascular network in our body or microarchitecture of complex organs such as the kidney and liver. So to overcome these challenges, we have been looking for a better way to build tissues with precision and reproducibility. To overcome this challenge, many investigators have sought for a way to utilize native tissue as a scaffold. This method has several advantages, including the preservation of microarchitecture and retention of bioactive factors within the extracellular matrix. This approach all starts with a donor organ. Cells in the donor organ is removed using detergents, preserving the tissue ECM that support the tissue framework. The decellularized scaffold is then repopulated with cells of interest. Using this approach, many organs have been decellularized and recellularized with target cells to develop an implantable constructs for functional restoration. I would like to share our experience in developing the kidney using this approach. Chronic kidney disease is a leading cause of death globally. As treatment is concerned, renal transplantation is the only definitive treatment for renal failure. Unfortunately, donor organ shortage has been a continued problem. So one possible solution is to use autologous renal cells on the decellularized organ scaffold to augment either partial or total renal function. In a clinical scenario, this approach involves taking tissue biopsies from a renal failure patient, isolate and grow kidney and vascular cells, repopulate the cells within the decellularized porcine kidney scaffold, then precondition in the incubator, followed by implantation back into the same patient where the cells came from. Donor kidneys are decellularized to remove all the cellular components, leaving only the structural frame with preserved vascular tree within the kidney. Once decellularization is complete, the vascular walls were uniformly coated with endothelial cells. This is the most critical step in the entire process, as any paucity in the coverage will result in thrombosis. In our studies, endothelial cell coverage was confirmed using imaging and histological analysis. We confirmed that endothelial cells cover the vascular walls of different caliber blood vessels within the kidney. The next step in the recellularization process is to seed the scaffold with target cells that would populate the scaffold to restore function, in this case, the kidney cells. We seeded the kidney cells in the kidney scaffold by injecting the cells into the kidney cortex. The individual single cells became organized into renal structures over time, as shown here. The formation of renal structures increased over time, as shown on this graph. The reconstituted renal structures express their cell-specific markers using various antibodies. The seeded renal cells proliferated as they formed renal structures. To determine whether the repopulated kidney cells are functional, we performed a variety of assays. We show that the renal cells are able to uptake electrolytes such as sodium. They are able to transport amino acids across the membrane. What is amazing is that the seeded cells produce erythropoietin, which is the hormone produced in the kidney. We then went on to test the feasibility of using the cell-seeded decellularized scaffold as a tissue construct to engineer kidney tissue in vivo. We used autologous kidney cells to repopulate the porcine scaffold and transplant it into the same pig where the cells came from. This slide shows the explanted kidney construct after 7 days of implantation. The gross finding shows normal-looking kidney without any visible blood clots. Histologically, we were able to find renal structures consisting of kidney cells using a series of renal cell-specific markers. This experiment demonstrates the feasibility of using tissue-derived scaffolds to engineer functional kidney tissue through the process of decellularization and recellularization. So in summary, we show that decellularization of porcine renal scaffolds can be achieved while preserving the tissue microarchitecture. Whole kidney engineering using the decellularization approach has advantages of preserving native tissue architecture and bioactive factors. These structural and functional outcomes suggest that implantation of repopulated kidney scaffolds may be an alternative treatment for renal failure. Another technology we have been developing for many years is bioprinting. This is due to the ability to deliver individual tissue elements, including multiple cell types, biomaterials, and biological factors and drugs to specific target locations in a layer-by-layer fashion to assemble three-dimensional tissue structures. We determined that the bioprinting technology can serve as a tool to build clinically relevant sized implantable tissues and organs with precision and reproducibility, which we could not do manually. Using a bioprinting system, our initial studies involved in bioprinting various inner tissue structures that mimic the native microarchitecture as you see on this slide. We showed that fine tissue structures can be replicated with corresponding cell and tissue functions. These microorganoids are currently being utilized for organ-on-a-chip applications using human cells to model the in vivo cellular environment. Some of the applications include drug screening and toxicology, tumor models for cancer diagnosis, and models for human development and diseases, and more. This currently is one of the hottest research areas in developing translational applications. To consistently manufacture the microphysiological systems with precision and reproducibility, we are applying the bioprinting technology to generate perfusible multi-tissue organoids with microchannels in one setting. This is currently an ongoing development which shows a great potential for many translational applications. On a larger scale, we have been developing various tissues and organ constructs for translational applications. This is an example of the processes we employed to develop muscle tissue constructs using a bioprinter. When designing a construct, one should consider the function of the tissue, in this case, the muscle. Since muscle generates unidirectional movement by contraction and relaxation of muscle fibers, we designed the construct using a unidirectional pattern to guide the cells to organize similar to native tissue, muscle. Since the muscle cells require sufficient nutrients and oxygen to function properly, we spaced each channel to maximize diffusion when implanted in vivo. The bioprinted muscle construct showed cell survival, muscle fiber formation, and neural integration in vitro. A rat tibialis anterior muscle defect model was created in one study, where approximately 40% of muscle tissue is removed in order to test the effectiveness of the bioprinted muscle construct. The implanted muscle construct matures into muscle tissue with neural integration, and recovery of muscle function was also confirmed over time. These are some of the tissues and organs we have been developing using the bioprinting technology with the goal of translating into the clinic. These range from soft to hard tissues and simple to more complex tissues. Most of these developments have been validated in preclinical animal models to demonstrate the formation of tissues and functions. Additional applications are on the horizon. So how do we want to use this technology for translation? We envision using the bioprinting technology in the clinic. When a patient requiring tissue reconstruction comes into the clinic, we can use the patient's medical data to determine the tissue needed for reconstruction. The data will also be used to reverse engineer a 3D CAD model of the tissue, and the code generated by the CAD/CAM system will then be used to instruct the printer to produce a tissue construct that could be used for surgical reconstruction. Of course, further development is necessary to bring the bioprinting to the clinic. One major challenge inherent to tissue engineering and surgical reconstruction is establishing adequate vascularization to integrate tissue implants into our body for survival and proper function. For many years, we have been developing approaches to sufficiently vascularize tissue engineered implants. Over the years, numerous ideas and concepts have been introduced and tested to improve vascularization to tissues, including the use of biological approaches such as growth factors and vascular cells to promote neovascularization. We have tried to incorporate oxygen-generating particles into scaffolds to provide oxygen to cells. Yet another interesting attempt involved the downregulation of cellular metabolism to conserve energy in the implanted cells. These approaches have shown some levels of success in preclinical studies. However, translating these concepts into actual applications remains to be seen, especially when dealing with large tissue mass. Recently, investigators have tried to pre-vascularize the tissue construct in order to accelerate vascularization. When a tissue construct is introduced, the body attempts to make new blood vessels from the periphery of the implant and infiltrate deeper toward the central core of the construct. However, if we create a tissue construct with vascularized channels, the host blood vessels would only need to connect to the pre-vascularized channel opening to perfuse readily, supplying the necessary oxygen and nutrients to cells within the tissue construct. Some of the physical approaches that have been developed include bioprinting, patterning of scaffolds to create vascular channels, and the casting method to recapitulate the vascular networks similar to native vessels. All of these approaches aim to prefabricate vascular channels to readily integrate with the host upon implantation. This slide shows an example of a perfusible vascular scaffold we have developed using the casting method that connects to native vessels in vivo. This method uses a donor organ, the kidney in this instance. In this example, a thermoplastic polymer, polycaprolactone or PCL, is infused into the renal artery to fill the vasculature throughout the kidney. Then the native tissue is dissolved completely in chemicals to leave a polymeric vascular casting, as you see here. The PCL vascular cast is then coated with collagen to create a shell over the vasculature. The PCL is then removed, which results in vascular scaffolds consisting of collagen with patent vascular channels that are identical to native blood vessels, which allow perfusion. Subsequently, the endothelial cells are seated within the vascular channel to prevent clotting, and the cells of interest within the scaffold, in this case, the kidney cells. The left top image shows the polymeric vascular tree after tissue removal. Under scanning electron microscopy, the microarchitecture of glomerulus is preserved with patent blood vessels that are perfusible, as you see here. The seeded endothelial cells cover the lumen of the blood vessel quite nicely. When implanted in vivo, the vascular channels integrated with host blood vessels. In this presentation, I have shared our experience in translating regenerative medicine technologies, and that this process involves complex steps that starts with the research and discovery phase to clinical trials and commercialization. Our goal is to bring effective treatments to patients. However, there are many practical and realistic items to consider. For example, even if a technology has been successfully developed in the laboratory setting, it does not automatically become a product. So successful discovery does not necessarily mean that the technology will be transferred to a manufactured product. Another important misconception researchers have is assuming that a product that addresses the clinical need will have a reserved seat in the market. Unfortunately, clinical needs do not equate to market needs. Similarly, scientific or clinical outcomes of a therapy does not guarantee the market success. We get excited when our developed product finally reaches patients through a phase 1 clinical trial. However, in reality, many phase 1 trials do not automatically advance to phase 2 trials. There may be many reasons for this, such as the trial outcomes, the cost involved in conducting the trial, weak market competitiveness. These are only a few considerations. There are many reasons for this, such as the trial outcomes, the cost involved in conducting the trial, and weak market competitiveness. Other translational considerations include understanding regional and global regulations and reimbursement of the therapy. These are only a few considerations. There are uncertainties and unexpected events as we develop products and technologies. One suggestion to minimize the effort and shorten the development time is to gain as much information as possible at the early stage of development. Specifically, it will be beneficial to engage experts from regulatory, clinical, and industry in the early stages of scientific discovery in order to know what to expect as development moves forward. To summarize, regenerative medicine has brought many tissue applications to patients. The translational research path is a complex process that requires a lengthy time, effort, and resources. Once a technology platform is developed, then that platform could be modified or applied in other clinical indications. And translational processes may be accelerated if manufacturing platforms are efficiently developed and regulatory processes are streamlined. Consistent with the report from the US Department of Health and Human Services, we believe that regenerative medicine will be the standard of care for replacing tissue and organ systems in the human body someday. So we're currently living in an exciting time to realize our goals. I would like to thank our Institute members for their devotion to develop regenerative medicine technologies and therapies to benefit patients. We're currently working on over 30 different tissues and organs, and there currently are 15 ongoing or planned clinical trials at our institute. I would also like to thank our sponsors who share the same vision as we do, that is to improve patient lives by developing regenerative medicine therapies and support technologies. Thank you for your attention. I hope this presentation provided you with a general perspective in the translational processes, including some of the considerations for developing a regenerative medicine product and how to best accelerate the processes. Again, I would like to thank Dr. Dongwoo Cho and Gina Zhang and the organizing team for the opportunity to share our experience in this symposium.